Chapter 5, Antibody Structure and Function. In this chapter, we're going to talk about the antibodies that are in the body and what their function is and all of the different types and things that we find. Antibodies are also called immunoglobulins. They're glycoproteins that are found in the serum, and we find them in the 5 major classes: IgG, M, A, D, and E. They all have a different function. So these are the key element of the humoral immune response. When we talk about the humoral response, we're talking about the elements that are found in the liquid portion of the blood, the plasma or serum per se. We talked about the cells and things earlier, but this is where we're going to talk about more of the plasma pieces of the body moving forward. Antibodies appear in the gamma band with serum electrophoresis. Now, if you don't know what this is, what we do is we take all the serum proteins in the body and we separate them based on their electrical charge and their size. Albumin would be the first one to travel through this gel, and he's really large with a very high negative charge. He stops relatively quickly. As you can see, this dark blue band here on the end. This creates a giant peak when we run it through a densitometer. On the end, we have this long gamma band. This is where all of the immunoglobulins are housed. If somebody was creating an abnormal amount of antibodies, we would see big peaks in this area. They could potentially have a monoclonal or polyclonal gammopathy that we'll get to later in the semester. But that is where those would show up, and that would be a screening tool that we would use to see if there is something like that going on. Here's the structure of an immunoglobulin. There's a basic 4-chain peptide with 2 heavy chains and 2 light chains, and they're held together by non-covalent forces and disulfide interchange bridges. We have variable and constant regions. The constant region is more of the bottom piece here. The part that's variable and makes immunoglobulins makes the antibody able to bind to different things is the variable region out here on the end. Okay, so that part's variable, the rest of this is constant. The bottom piece is called the Fc fragment. It's got no antigen binding ability. It's held together by sulfide-sulfide bonding, and it's important in effector functions of immunoglobulin molecules such as opsonization and complement fixation that we're going to talk about during week 4. So I'm not going to get into a lot of detail about that right now. This is the part that could potentially be bound to some type of a lymphocyte. The Fab region is held together by disulfide bonding. So this Fab region I call Fab hands or jazz hands. I always think of it looks like somebody standing with their hands up in the air doing Fab hands. That is what grabs on, those hands grab onto the epitope of an antigen. So that is where the antibody binds to the foreign organism or whatever it may be that it's binding to. This is held together by disulfide bonding. It's obtained by papain digestion of an immunoglobulin, and the end is the that antigen binding area. So Fab binds to the epitope. Light chains are something that were discovered in 1854 by Henry Bence Jones. Now the clinical significance of this that I want you to know is that they are light chains, and it becomes a kind of a cancerous situation where B cells get a mind of their own and they start spewing out boatloads of abnormal immunoglobulins, and we find something called Bence-Jones protein, especially in their urine. So if somebody has abnormal amounts of protein in their urine, usually we can go on and do electrophoresis of their urine to see if this is present. If they do, it's indicative of multiple myeloma, which is a plasma cell disorder. That is incredibly important. The heavy chains of the immunoglobulin have a variable and constant region that I just mentioned before, with different carbohydrate sequences on them, designated as CH1, CH2, and CH3. I don't expect you to have to know a lot about those. Just know there are carbohydrate sections on them that designate the constant regions. And they are unique to each class and give the immunoglobulin type its name. So here's a better picture of what I'm talking about. It includes a little bit of everything. So we got the antigen binding site, which is also the Fab region. We got the Fc region, which I consider the stalk. It looks like the stalk of the tree. We got the carbohydrates here on the sides. We've got the hinge in the middle that's got the sulfide-sulfide bonding there. And you can see that this is typically what an immunoglobulin or IgG1 might look like. Okay. Now, an IgA kind of looks like a crab walking through the sand. And IgM is very large. It's actually got 5 immunoglobulins stuck together, held together by a J chain. So there's different types of heavy chains. We got isotypes, allotypes, and idiotypes. You need to be familiar with the definitions of each. An isotype heavy chain is a unique amino acid sequence that is common to all immunoglobulins of a given class in a given species. The allotype has minor variations of the isotype sequences that are present in all individuals, in some individuals, but not others. And the idiotype is variations in variable regions that give the individual antibody molecules its own specificity. We talked about the hinge briefly. Okay, we got this in the middle. It allows those arms to be flexible. I think of those little flexi guys that are filled with air that you see on the street corners trying to advertise something, and they kind of flop around, and how those arms are very flexible. That's kind of how this works. They can flex and bend to bind certain antigens. The carbohydrate portion here is found on all of them. Okay, you can see the carbohydrates on the side. We saw it called oligosaccharides in the last couple slides. But this increases the solubility of the immunoglobulin, so it can actually float around within the plasma. It provides protection against degradation, and it enhances the functional activity of the FC domains as well. The first one we're going to talk about is IgG. This is the predominant immunoglobulin in humans. It's 75 to 80% of the total serum immunoglobulins. It's got the longest half-life, so it lives quite a long time. And there's 4 subclasses. 1 through 4, you can see number 1 is in the highest prevalence. The major functions include providing immunity for a newborn. IgG can cross the placenta, so it can go from the mom to the baby in utero. It's got the ability to fix complement that we're going to learn about next week. It coats an antigen for enhanced phagocytosis or opsonization, and it neutralizes toxins and viruses, and it participates in agglutination and precipitation reactions that we'll be doing later on in the semester. IgM is known as a macroglobulin. He is so strong, he can react at room temperature, where usually things like IgG that we went through react at 37 degrees, which is body temperature. He can bind up a lot of immunoglobulin, even though he only accounts for 5 to 10% of the total serum immunoglobulins. He's got a half-life of 6 days, which is pretty short, and he can exist as a monomer on the surface of B cells or a pentamer, which is what you see in the picture here, found in secretions. He's got J chains that hold him together. It's kind of like a star-like pattern, I guess. This one cannot cross the placenta. It's known as the primary response antibody. So if you think of somebody getting sick with the chickenpox for the first time somebody is exposed to the chickenpox, their body will have kind of a longer, like a lag phase, and I'll show you a picture of this here in a minute. But the very first thing that's going to come into play the very first time you get exposed to something is IgM. He's the major player in that primary immune response where you're first exposed. Okay? He's synthesized only as long as the antigen remains present. He's very good at complement fixation because he can bind a lot of stuff. So here's what I meant about that primary versus secondary response. So when you're first exposed to chickenpox, as you can see here in the beginning, you've got this longer leg area. Then we have this incline where IgM comes up, and IgG comes in a little bit later. The second time you'd be exposed to chickenpox, you have a much shorter leg and a much higher, 1,000 times greater response of IgG over IgM. So IgG is more of that memory response that comes in later, where IgM is usually involved more in the primary response. The next one is IgA. This one accounts for 10 to 15% of all circulating immunoglobulins, and it patrols mucosal surfaces and acts as the first line of defense. This one is found in your tears and saliva in your body. So it's able to neutralize those toxins produced by microorganisms, and it prevents bacteria from adhering to those mucosal surfaces. It does passively transfer immunity to the newborn as well. So that's what you really need to know about this one. It does also have a J chain. Secretory IgG is also found in plasma cells, mainly in the mucosal-associated lymphoid tissue, or the MALT. It's released in a dimeric form, and it's captured by a secretory component on epithelial cells. IgD is the one that's extremely scarce in serum. It's less than 0.001% of all immunoglobulins. It's very susceptible to proteolysis and has a very short half-life of 1 to 3 days. This one we find on the surface of the immunocompetent but unstimulated B cell lymphocytes. You can see it sitting on top of a B cell here. It plays a small role in B cell activation and in the regulation of B cell maturation and differentiation as well. The last one is IgE. This one is very, very, very scarce, but quite powerful actually. It's 0.0005% of the total serum immunoglobulins. It's produced by plasma cells that are located primarily in the lungs and the skin, and attaches to basophils, eosinophils, and tissue mast cells. There is a cascade of events that results in the degranulation of mast cells that release things like histamine and heparin. If you think of histamine, that is something that makes you itchy and sneezy. So you take an antihistamine that you get from the drugstore to kind of stop that sensation from happening when you get like seasonal allergies per se. Another type of IgE function is allergic reactions. We see this in a type 1 immediate hypersensitivity reaction. Things like hay fever, asthma, vomiting, diarrhea, hives, and other life-threatening anaphylactic shock type situations would be where this might come from. And a lot of us do struggle with seasonal allergies, so it sounds familiar, right? Another IgE function is parasitic infections. Eosinophils will play a role in the destruction of large antigens such as parasitic worms that cannot be easily phagocytized. You can see this guy here with a worm coming out of his foot. The last piece we're going to talk about is monoclonal antibodies. We use monoclonal antibodies all the time in the laboratory for diagnostic testing and therapeutic purposes. So, for example, most of you have taken microbiology already, and if you have, you may have used a staph latex to look for something like staph bacteria if you're not sure if it's a staph or a strep. So if you add that little drop of latex, it's got antibodies connected to latex beads that will attach to that staph. These were produced to be used in those test kits as monoclonal antibodies. Every antibody in that test kit is identical. So they're developed based on the knowledge that B cells are genetically pre-programmed to synthesize a very specific antibody. They're also derived from a single parent antibody producing a cell that has reproduced many times. So what they do is they inject the mouse with the antigen. They use a plasma cell. The mouse's plasma cells produce some antibodies. They then use tumor cells to create a hybridoma that creates a monoclonal antibody. I'm not going to get into more details on this, and I know I usually get a lot of questions on it. If you want exact details as far as what exactly is going on in these cells, you can certainly find a lot more information online. And I'll try and find a reputable resource for you that I'll add to your course as extra information. You don't need to go into big detail on that, but a lot of people do become curious on how this is done. So I will get that in there for you. All right, that concludes our section on immunoglobulins.